Okay, so we'll see the three domain system and kingdom classification. So the domains were Archaea, Eubacteria and Eukarya. Okay, Archaea, Eubacteria and Eukarya. And what were the six kingdom classification? Those were Archaeobacteria, Eubacteria, then all these remaining Protista, fungi, plantae and animalia. Okay, so what led to this three domain system or kingdom? Okay, before that you need to understand that domain is bigger than kingdom. Okay, we have learned the hierarchy level. Okay, so it's kingdom, the phylum, class, order, how it goes. It is above kingdom, domain, a bigger category. So what happened earlier, Monera was the kingdom in which all the bacteria were the sole members of this particular kingdom. But when studies were happening, what did happen? That he found, Carl found more a type of bacteria which had the capacity to survive in extreme conditions, extreme environmental conditions, and it had cell wall composition which was not similar to eukaryotes or any bacteria. It had a very different structural RNA, like the RNA was very different from that of eukaryotes or other bacteria. Okay, so you see how the depth of study is going. Earlier, it was just morphology. Then we included cell wall, cell type, mode of nutrition. Then see at this, we have reached at the RNA study level synthesis. The difference in RNA created a different domain of bacteria, okay, which is known as archaea. Okay, so for example, the bacteria which can survive in very hot conditions, very high temperature conditions, it is called as thermophiles. Thermophiles. Okay, in acidic and in high temperature, we call it as acidothermophiles. Okay, so in extreme climatic conditions, so you need to understand, if they are surviving in such extreme climatic conditions, their composition has to be different from a common bacteria or a different eukaryote, so that they can survive in that particular extreme conditions. Then in eubacteria, all the bacterial organisms like bacteria were included under this domain. So earlier, both these bacteria was in one kingdom, Monera. So that is why only the 5 kingdom classification was made. But then later on it was seen that the characteristics in Archaeobacteria is really different to be included in one kingdom. Okay, so that is why a different kingdom altogether was created, which only included Archaeobacteria, the bacteria which can survive in extreme environmental conditions. Okay, then Eubacteria, the bacteria that survive in normal conditions. Like you all know, the bacteria have certain optimum temperature, optimum pH for their functioning to purpose. Now, any organism, any functioning in any organism, if it has to happen, it has to happen in optimum conditions. Okay, in a particular temperature, in particular pH, all those things has to be there so that for the organisms to survive. So U word, when it is there, it is true bacteria. Okay, then further all the other eukaryotes which are there, they were categorized into the other 4 kingdoms. And under eukaryotes, we have unicellular, multicellular, whether it is autotrophic, heterotrophic, if it has cell wall or not, the composition and everything will be included when these kingdoms were made. Okay. So in this chapter, we will be learning the 5 kingdom classification, because the idea behind this chapter is to understand the process of classification, how it was done, or the basic characteristics of each member in the kingdom. Okay. So I hope you all understood the difference between 5 kingdom classification and 6 kingdom classification, and what is a domain, and how it led to the creation of these 3 domain. And the eukarya domain includes all the eukaryotes. Okay. So further, the Monera kingdom, we'll start with Monera kingdom in the next part of the video. Okay, so as we started with kingdom Monera, like we just discussed where does kingdom Monera and all belong to. We discussed the 3 domain system, 6 kingdoms classification, and even 5 kingdom classification. As I had said, in this chapter, we will be dealing with the Whittaker system of classification, that is the 5 kingdom classification. So that is why we will be starting with kingdom Monera. Okay. So whenever the word Monera comes, you have to remember the sole members or the basic members of kingdom Monera is bacteria. Okay. As we had discussed that initially all type of bacteria, that is the bacteria that live in extreme climatic condition, bacteria that live in optimum climatic conditions, all were grouped under one kingdom. Later on, it was categorized into different domain. So the ultimate idea or point is bacteria are the sole members of kingdom Monera. Then as you all know, kingdom is the highest category, so there will be more subgroups inside it. Okay, so one of the subgroups that has been created, there are 4 categories on the basis of their shape. Okay, so whenever any categorization or grouping is done, it is always on the basis of certain characteristics. So the 4 categories which we are going to discuss is on the basis of shape. Okay, on the basis of shape. First is coccus, a group of bacteria which will be named as coccus. I'll be saying the singular one, plural will be mentioned afterwards. So coccus, which means spherical in shape. Spherical in shape. Then bacillus. Bacillus meaning rod shaped. Vibrium means comma shaped, and spirillum means, the word itself tells you, it is in spiral shaped. So you can see how the categorization is done. It is done on the basis of its shape. Okay, so the naming which is there, that is done. These were the 4 categories that were made on the basis of shape. Coccus being spherical in shape, bacillus in rod shape, vibrium comma shaped, and spirillum spiral shaped. So whenever a bacteria is named, depending upon its shape, the name like bacillus is added. For example, if I can give you lactobacillus, the bacteria that is found in milk, lactobacillus. So what you can make out from the word, that the particular bacteria is in rod shaped, and even the suffix prefix has a meaning to it. Okay, so lactobacillus, bacillus gives you shape, and lactose is the substrate on which this particular bacteria will act upon. Okay, so we'll learn in detail afterwards the examples. As I had said, I will be including all the examples in the presentation, okay, with the images and everything. So don't worry about that. Now, bacterial structure and behavior. So how is the bacterial structure? It is simple. Okay, because as we all know, it is a primitive organism, the structure is gonna be simple. But that does not mean the behavior or the functioning of the bacteria is as simple as its structure. No, it is not. The functioning or the behavior is complex. As you all know, because the bacteria, some of the bacteria have the capability to switch their mechanism of reproduction. They even have the capacity to come up with functioning inside their body so they can survive in the unfavorable climatic conditions. So that is how their behavior or the functioning becomes complex. Okay. Then Archaebacteria, eubacteria, as I had mentioned before. Since we are learning Whittaker system of classification, we will be learning Archaebacteria and eubacteria under kingdom Monera. Okay. So Archaebacteria is the group of bacteria that can survive in extreme climatic conditions, extreme environmental conditions. For example, if there are group of bacteria that can survive in high temperature, they are called as thermophiles. They are called as thermophiles. If there are group of bacteria that can grow in high salinity, then they are called as halophiles. And many more terms I like that. For if some group of bacteria can grow in high acidic medium, then they are called as acidophiles. Okay. So these are the group of bacteria. So just imagine if they can survive in such extreme climatic conditions, you need to understand, students, that their structure or their functioning or mechanism which is there, it is going to be different when compared to a normal bacteria or a true bacteria. Okay, their RNA is different, their cell wall composition is different, even their mechanisms are complex, so that it helps them, whatever functioning which is there, which is different, that helps them to survive in the particular climatic condition. Okay, example they have given is methanogen. Okay, methanogen is a group of bacteria that can, I'll write down here. Methanogen, it's a group of bacteria that is found in the gut of the ruminant animals. I hope you all know what ruminant animals are. They are the ones that first swallow in the food, bring back the food back into their mouth and ruminate and churn their food again. So those animals that does like that, they are called as rumination. The process is known as rumination. So imagine the gut will be having different types of enzymes, right? For many various purposes inside the body. So the bacteria, if they can survive in that particular gut, they have to be archaebacteria so that they can survive in that particular condition. So methanogens are basically, if you all know the cow dung which is there, it is used for biogas production. Okay, methane gas production. Now, how is it possible? It was possible because of the presence of methanogen. Okay, so this question is usually asked when asked about archaebacteria, that what does methanogen's function is, and methanogen, where does it belong to, whether it is belonging to archaebacteria or eubacteria. Okay, so then comes eubacteria, the true bacteria that has to have some optimal conditions to survive. Like they cannot, if for example, if their optimum pH is 8 or 6, if their optimum pH, I'm just giving you a example. If the pH varies, whether it becomes less or more, the bacteria won't be able to survive. Okay, such group of bacteria is known as eubacteria, U standing for true bacteria. Okay, so the example they have used is cyanobacteria. Since there are many points to be covered in eubacteria, we'll come back with the topic eubacteria.